So, okay, welcome everybody. We are here today. We are going to be in a very familiar moment. So we have the pleasure to have here Ava Garrison. She is a postdoc researcher at the Michigan State University. She is here visiting the Thaliana Biological Station at the Chavi Pico lab. So the floor is yours, Ava.
Thank you. Thank you so much for having me, and thanks to everybody who was able to make it today. I know there's a lot going on at the station, so I appreciate you being here. And yeah, as you mentioned, I'm from Michigan State University, but I'm also from specifically the Kellogg Biological Station, our own biological station at MSU, where we do a lot of evolution and ecology research. So really excited to be here today to present some of it to you guys. And I'm representing the Connor Lab at the Kellogg Biological Station. So I'll be presenting today some work on the evolution of trait loss along altitudinal and latitudinal gradients. And I'll start really broadly by saying I'm interested in adaptations, which are the traits that function to help an organism deal with challenges in the environment or utilize opportunities in the environment. So there's some really cool examples in nature, like the ability of cuttlefish to camouflage so effortlessly. Pretty much everything about the platypus, from its aquatic adaptations to venom to electrolocation. And then for flowers, just utilizing pollinators to overcome their lack of any inability to self-pollinate. They can attract pollinators by having these really intricate designs in their flowers. But adaptation is everywhere. All life on Earth depends on adaptation. So in addition to these really cool examples, there's examples in pretty much everything we see in nature. So it's really important to study adaptations and the mechanism behind adaptations. It's also interesting to think about what happens when the environment changes and an adaptation loses a function. What happens then? So there we typically see trait loss. So when a trait is no longer functional, we expect that the trait will be lost. And there's some really cool examples of that. One being eye loss in cave fish. So this has evolved multiple times when fish populations near the surface have migrated into more cave waters with low light, and their eyes are no longer functional, so they completely lose their eyes. This is a pretty extreme example of trait loss, but a really cool one. So we expect when there's adequate variation for trait loss and strong selection, selection can come from the cost of the trait itself, or perhaps there was a trade-off with the trait that one half of it is now, it's non-functional, it's not worth the trade-off. So the trait loss can be rapid in those cases. Sometimes though, a trait can be maintained even if it's seemingly non-functional. And there are three non-mutually exclusive mechanisms to explain the maintenance of non-functional traits. The first is drift, so genetic drift. When there's a lack of genetic variation, trait loss can be constrained if there's not adequate genetic variation to lose that trait. This is the opposite of what we usually see in genetic drift, where random variation, random fluctuations in allele frequencies lead to trait loss. In this case, we're actually seeing unwanted trait maintenance due to lack of genetic variation. Another example is pleiotropy. So genes that underlie the trait of interest may also influence another trait, and this trait could be beneficial, and that relationship could be what's maintaining this less beneficial or non-functional trait. And then finally, it could be selection. So maybe it's not functional in the way we expect, but there could be some other function to the trait, or a function that we're not seeing in the trait that could be maintaining the trait in the population. So those are some ways it can be maintained. And what I'm going to switch my focus now to is a system in which we have a really great opportunity for understanding these mechanisms. So I'll switch focus to Brassicaceae. So hopefully we have some people that like plants, but if not, I'll try to keep it interesting for everybody. So Brassicaceae is a really important plant family. A lot of the crops that we eat, broccoli, kale, things like that, come from Brassicaceae. Plus a lot of crop weeds and invasive species are from the family Brassicaceae. So it's a very important family to study in ecology and evolution. What else is interesting about Brassicaceae is that it has this diagnostic trait called tetradynamy. So tetradynamy is when you have 4 of these long stamens. So stamens for non-plant people are what produces the pollen. And then 2 short stamens. So this is present in most species in the family Brassicaceae. And there are some ideas for why tetradynamy may exist and persist in the family Brassicaceae. So it's known that these short stamens are actually producing more pollen than the long stamens. So pollen from the long stamens are removed by pollinators earlier. And so these short stamens could potentially be sort of a backup for pollinators. Another hypothesis that's being investigated by a student in my lab, Robin Waterman, is the movement hypothesis. So this is a new idea that she has been working with, where movement of pollinators between the long and short stamens is actually increasing your chances of pollen being picked up and deposited on the stigma, which is where what accepts the pollen for reproduction. So in this scenario, you could have a pollinator feeding on long stamens while making contact with the short stamens, and then they move around to feed on the short stamens, and the pollen is deposited on the stigma. So that's one hypothesis for the maintenance of this heterodynamy, this weird two short and four long stamen that we see all over Brassicaceae. But my question is, what happens for plants that self-pollinate, that don't require a pollinator, and this movement hypothesis or even the backup pollen hypothesis wouldn't pertain to it? An example of that would be Arabidopsis thaliana. So Arabidopsis transitioned to selfing many, many centuries ago, thousands of years, I would say. And so we expect that there's no longer a function to these short stamens, and so we would expect to see trait loss. So there has been studies in the lab from previous students, Anne Royer, who looked at short stamen function. So Anne performed these stamen removal experiments in growth chambers, where she, in the bud, preferentially removed stamens, either short, long, or both, to end up with flowers with 0 short stamens, or 0 stamens at all once they open, only short stamens remaining, only long stamens remaining, or all stamens remaining. And what she found was that there was no function to be seen from the short stamens in pollination. So plants that had only short stamens remaining, or flowers that had only short stamens remaining, produced as many seeds per fruit as flowers with 0 stamens remaining. So essentially no selfing ability in these short stamens, whereas plants with long stamens could produce as great a seed set as flowers with all stamens present. This was followed up by a student, an undergraduate student in the lab, Kola, who looked at natural variation stamen number. So plants with, flowers with no short stamens, 1 short stamen, or 2 short stamens. And he found that there was no seed set difference in these naturally occurring variants in short stamen number. So again, we're not seeing any added benefit of having these short stamens. They're not functioning in selfing. So what's the point? With this, we would expect to see short stamen loss in Arabidopsis thaliana, and indeed, we do have evidence of ongoing short stamen loss in Arabidopsis thaliana. So as I mentioned, Arabidopsis is selfing, and it is quite selfing. There have been reports of less than 1% outcrossing rates in Arabidopsis thaliana, so it is mainly just selfing and not relying on outcrossers at all. And we can see in these variants, we have these 0 short stamen flowers, 1 short stamen flowers, and 2 short stamen flowers. The short stamens are lost completely. There's not smaller short stamens, they're gone. They're not partially removed, they're just fully gone. We do see variations of that somewhat, but for the most part, they're completely gone. We don't, however, typically ever see the long stamens missing from the flowers. It's just the shorts that we're seeing lost on a regular basis. So these long stamens, when they're absent, are long enough that they're able to self-pollinate. They're reaching the point of the stigma where the pollen is deposited for reproduction. So they're all that's necessary for reproduction. And the short stamens, when they're present, have no function, as we saw from the previous data. They're simply too short to reach the stigma and not functional. So it's expected that we're seeing trait loss for short stamen number. But what's interesting is that it's incomplete, and it's incomplete in a pretty specific way. So we're seeing that at high latitudes, populations are maintaining more short stamens, so they have more plants with more 2 short stamen flowers and 1 short stamen flowers versus 0 short stamen flowers. But for the most part, these populations have mainly 2 short stamen flowers. But at low latitudes, we're seeing the opposite. So we're seeing more plants with lots of 0 short stamen flowers and 1 short stamen flowers versus flowers with all the short stamens intact. So this latitudinal cline has been found, as well as multiple altitudinal clines. So in the Pyrenees and South Spain, populations that Xavier has given to us and has been working with, we find the same pattern with high altitudes, more maintenance of short stamens, so less loss of the trait, where at low altitudes we're seeing less maintenance, more loss of these short stamens. So we are interested in the reason for this maintenance, and there's the 3 hypotheses I brought up earlier. There could be drift, so at these higher latitudes and altitudes we could have lower genetic variation, maybe from lower population sizes that are constraining the loss of this trait. There could be pleiotropy, so genes underlying stamen number could also be influencing traits that are adapted or that are conferring adaptations to high latitudes or altitudes, like cold or high sunlight, something like that. We're still working on that. And then finally, there could be selection. So there could be different function of short stamens at high latitudes and altitudes compared to low latitudes and altitudes, and that could be the reason why these short stamens are maintained. So our lab is taking several experimental approaches to discover these mechanisms underlying the maintenance of these short stamens at high latitudes and altitudes. So today I'll be going through some of those experiments that we've been working on and what we have planned for the future. So for genetic drift, previous and current work in the lab being done is looking at nucleotide diversity along an altitudinal cline to look for evidence of drift constraining trait loss. We're also investigating pleiotropy using a chamber common garden experiment. So we're looking for traits that co-vary with short stamen number along these latitudinal and altitudinal gradients that could be the reason why these short stamens are maintained. And finally, selection. So the reason I'm in Spain is to do these field studies at high and low altitudes to look for differences in short stamen function at different altitudes. And so I'll go through that and then show you a little bit of future work that we have planned in the lab. But to start, I'll go over this study on nucleotide diversity along an altitudinal cline. And so this work was done by a student in our lab, Sophie Bicey, along with collaboration with Xavier Picot. Very cool work being done, and it has a paper currently accepted pending minor revisions and evolution, so very exciting there. But what they found was that nucleotide diversity, they measured nucleotide diversity along an altitudinal cline. So we have this cline in the Pyrenees. These are just population codes for populations around the Pyrenees. And it's been found here and in previous places that there is a cline in short stamen number with more short stamen numbers, more short stamens at higher elevations. And this paper also found that nucleotide diversity is lower at these higher elevations. So there are basically matching clines but in opposite directions where nucleotide diversity is lower at high altitude and short stamen number is higher. So this lower nucleotide diversity is likely due to smaller affected population sizes. This is likely due to founder effects from recolonization of these populations. So this has been found in the past and is somewhat expected, but the question here is whether this pattern, whether this drift is actually something that's explaining decline in short stamen number. So is drift responsible for this pattern of more short stamen numbers at higher elevations? So there was a multiple regression done that included elevation, short stamen number, and nucleotide diversity. And what was found is that the altitudinal cline in short stamen number is still significant, even when accounting for drift. So that is telling us that the cline is present despite drift, meaning drift is likely not the thing that's constraining loss at these high elevations. Other factors must be involved. So that is lessening drift in our list of potential mechanisms because the cline is present despite including the effects of drift on this altitudinal cline. So from there, I'll move on to the next experiment that we have working on in the lab, and that is this growth chamber common garden. So for this, we're using lots of different populations across the range of Arabidopsis thaliana. So we have two different sets of populations that we're using. The first is this broad set in green, and that is using populations from the 1001 Genomes Project that Arabidopsis, has a lot of Arabidopsis data available for. And it is a very wide distribution, but there is little replication. So we have 214 populations, but only 253 lines. So most of these populations, we have one line per population. So wide distribution, little replication. On the other hand, we have this intensive set where we look at basically the opposite thing. We have fewer populations, but a lot greater replication per population. So we have 720 lines total with about 5.8 lines per population on average. And this is focusing more on the extremes of the altitudinal latitudinal gradient. So there's more replication among these altitudinal gradients and more populations on the extremes of the latitudinal gradient. And these populations were collected by evolutionary biologists, mostly collaborators. So we have a lot of data about these populations that we can use and a lot of confidence in the collection of these populations. So using these two populations, I'll show you, or these two sets of populations, I'll show you what we have going on in the lab. The broad, I should say, has been completed, and the intensive is ongoing. So the intensive, since there's so many populations, we're breaking it up into several groups. The third group is happening in the lab currently in the growth chambers. So in multiple groups, we have these plants grown in a common garden at the Kellogg Biological Station, where we measure a large suite of traits. So we have morphological traits, flower size and different dimensions of different floral traits, leaf number, plant height at flowering, plant height at the end of life, ovule number, things like that, as well as phenological traits, so flowering time, bolting time, time to stop flowering, so flowering duration, things like that. And the point of taking all these traits is to look for traits that co-vary with short stamen number across latitudes and altitudes in order to find evidence of pleiotropy. So when we see traits that co-vary and also have an effect on short stamen number, that is evidence of pleiotropy. So I'll show you the results from the broad set first. As I said, that is complete, so we have more data from that. 1 minute. And this work was done, or analyzed mainly by a former master's student in the lab, Charlie Anker. And she found first that the latitudinal cline is replicated. So using this large distribution of chamber plants, she found that indeed at higher latitudes we still see more short stamens than at lower latitudes where we see more loss of short stamens. And the altitudinal cline is regional. So if you focus on the Mediterranean populations only, we do see a cline of more short stamens at high elevations and fewer at low elevations. We were not able to replicate this cline anywhere else in the region. It seems the Pyrenees and the southern Spain populations and one other mountain range in central Spain seem to be the only places that have these clines that we can find so far, but we're still looking for other populations where the cline might be repeated. But what we found so far, or what we're looking for, are traits that explain the short stamen number clines. So those traits need to be also have a cline altitudinally or latitudinally. In this case, we just looked at latitude because it encompassed the whole set. And that trait would also need to predict short stamen number after correcting for latitude. So there had to be a regression on latitude as well as on short stamen number. So first, we regressed the traits on latitude, and 3 of the 18 that we measured showed clines. So of course, short stamen number showed a strong cline for latitude, and the other 2 traits we found were flowering time, so from germination to flowering, how long did that take, and leaf number. Leaf number was measured at bolting, and it's sort of an indication of how quickly it's growing. The plants that grow very fast tend to have fewer leaves at bolting than the plants that take a longer time, but it could also be to do with rosette density and stuff like that. So there could be multiple things contributing to leaf number. What we found though when we regressed these traits on short stamen while including latitude and correcting for latitude is that neither leaf number nor flowering time predicted short stamen number after controlling for latitude. So that tells us that in this broad set, we have not identified any traits that may be contributing to the retention of short stamens via pleiotropy. So next, I'll show you the preliminary results from our intensive set. Again, this is a smaller distribution, but more replication. What we found is, again, these latitudinal clines are replicated, same pattern, greater short stamen number at higher latitudes, and within the Mediterranean, greater short stamen number at higher altitudes. So with our more intensive set, the same pattern is there. We are still working on taking data and analyzing our different traits, but what we found from a preliminary analysis, regressing the traits we have on short stamen number, we did identify flowering rate as having an effect on short stamen number while correcting for altitude and latitude. So this is evidence or potential evidence that pleiotropy with flowering rate is possible. So we find that at low altitudes, rate tends to be faster, and low latitudes and altitudes, they tend to be faster flowering rate. And we're seeing this pattern co-vary with short stamen number. And so this could be explaining the maintenance of short stamen number if it's beneficial to have this rate be consistent or vary across altitudes and latitudes, and it's tied to short stamen number. That could be one explanation. I'll move on now to our final hypothesis, which is selection, and that's the work I'm doing here in Spain. So we started this last year with G.B. Bingman, another grad student in the Connor Lab, using two sites, one at low altitude, the El Castillejo Botanical Garden, and one at high altitude, the Hoya de Pedraza Botanical Garden. And we have populations from around Spain, focusing on low altitude and high altitude populations from around Spain. So we had 9 populations total with 193 plants last year. And this was all done with collaboration from Javier Picot, who set everything up, and we would be nowhere without him, so we're really appreciative for this collaboration. And you can see there's some clear differences. At the high altitudes, we're seeing a lot more snow cover versus the low altitudes, which are warmer and starting earlier generally. So we did 3 experiments at these field sites last year to determine short stamen function as well as short stamen cost. So stamen removal and natural variation surveys. This is the same as we saw earlier in the talk done in the lab, where stamens were manipulated in the bud, removing either the shorts, the longs, all of them or none of them, to look for short stamen function, and as well as natural variation, where we just find short flowers with 0, 1, or 2 short stamens and look for seed set there. One other point is that these stamen removals done at each site were restricted to populations from matching altitude. So at the low site, we did experimental manipulations on plants from low altitudes, and at the high site, we did manipulations on plants from high altitudes. This was to look for function in their own native environment. Even though they're from all across Spain, we're matching altitudes to mimic their home environment. And then we also looked, we did a pollen addition experiment, so hand pollination of these flowers, and this is to look for the cost of short stamens. So the idea is if we're providing ample pollen to these flowers, we can see if the total seed set is higher with 0 short stamens, which would indicate that producing those 2 short stamens is having some cost to the total seed set per fruit. So if you make 0 short stamens, but you have all the pollen you need, and you make more seeds, that suggests that short stamens have some cost to production that we're able to actually quantify. I mean, it's expected that they have some cost because they're a structure, they're like a 10% of the flower, but it's difficult to find costs in nature. So I'll start by showing results from our stamen removals at the low altitude. So this is similar to what we saw last time. We have stamen removals in the bud with either all removed, so nothing left, only short stamens left, only long stamens left, and then all stamens left. But we have 2 treatments here. One is the sham, where we go in the bud, jostle it, and then do nothing. And then one where we just find an open flower with 2 short stamens and mark it. So we can include the cost of messing with the bud. And then we're looking again at mean seed production. And as we saw with the chamber work previously, there's no evidence of short stamen function. So the plants, the flowers with 0 and 2 short stamens, 0 stamens and only short stamens are making no seeds per fruit. Whereas if you have 2 long stamens, if you have 4 long stamens, you're making essentially as much seeds per fruit as a plant with all stamens present. And then of course, if you were not messed with as a bud, you did best, but that was to be expected. So at the high site though, we see something different. These flowers with only short stamens remaining are actually making a non-zero amount of seeds per fruit. It's still far less than what we're seeing with 2 or all stamens present, long stamens or all stamens present, but the fact that we're seeing a non-zero amount of seeds is evidence of higher function of these short stamens at high altitudes. So this is a cool result that we found last year that we're excited to look into more. However, when we looked at just variation at either site, so this is again what we saw previously, where we have plants that naturally have 0 short stamens, 1 short stamen, or 2 short stamens. We looked at that at low and high sites for just mean seed production per fruit. There was, like we saw last time, no difference in seed set per fruit by short stamen number at either site. So you have no greater seed set if you make 0 short stamens than if you make 2 short stamens naturally. So this is suggesting that despite evidence of greater function of the short stamens at high altitude from the stamen removal experiment, in naturally occurring flowers, it doesn't really seem to benefit having 2 short stamens at either site. And if anything, it seems like having 0 short stamens may lead to higher seed set. This is clearly not significant, but there is a hint that there could be a higher seed set for plants with 0 short stamens, which could suggest a cost of producing these 2 short stamens. So that's what this last experiment, this pollen addition experiment, is testing. Again, we have the low site and high site, and we have added pollen to flowers with 0 short stamens naturally and 2 short stamens naturally. And again, we're looking for mean seed production. And as I said, we'd expect to find higher seed set in these 0 short stamen flowers once we've saturated them with pollen, if there was a cost associated with producing short stamens. But we found no difference in seed set at either site, no significant difference, and no overall difference either. We have this weird pattern here where maybe there's greater fitness in 2 short stamen plants at the low site, and maybe there's greater fitness in 0 short stamen plants at the high site. It's hard to explain, but overall we're seeing no significant cost associated with these short stamens. We also see that at the high altitude, we're seeing somewhat higher seed set. It's not significant here. It was on the natural variation slide, and that is likely due to the fact that we had different populations present at the 2 sites. But perhaps the ones at the high site tend to make more seeds per fruit, generally have more ovules per pistil, perhaps. So that could explain one of the differences we see. But for this year, we're repeating the experiments with a larger sample size. So we have about more than double the plants from last time. And we have populations repeated at each site. So that will take away any between site differences that are just due to populations and not due to the sites themselves. And will also give us data on plasticity. So if there's some different function between the 2 sites that is due to plasticity within the environment, so maybe at higher latitudes, the short stamens could be longer for some reason, that are better able to pollinate. We don't know. But we could see if there's plasticity by repeating these populations at each site. So that's one thing we have going on. And then we're also taking a lot more data. So we have stamen count averages for each plant at each site. So we can also look for differences, environmental differences in short stamen number in case there's plasticity there, who knows. We're also doing morphology measurements. So flower, with these pictures, we have information on stamen length and closeness to the stigma. And then we're also taking data on phenology, so flowering time, duration of flowering, and then we're taking overall fitness at the end. So a lot of cool data we'll be taking currently, and new results coming soon that I'll be excited to share when that's done. So to wrap up what I've talked about so far, we have investigated these 3 hypotheses somewhat. So for genetic drift, we're not seeing significant evidence that that is causing the altitudinal climb. It doesn't seem like these high altitude populations are being constrained in short stamen number, and that's what's maintaining them. And if there is any constraint, it's likely not the only mechanism. For pleiotropy, we have some preliminary evidence that perhaps flowering rate could at least partly explain a latitudinal climb in short stamen number, but we're still analyzing those, we're still collecting those data and then analyzing them. So we'll have more for that soon, hopefully. And then for the field study, we have no evidence of cost, but there is evidence of greater function. So this could help explain greater function at high altitudes. So this could help to explain the altitudinal climb. So cool results so far, but a lot still ongoing. So real quickly, I'll talk about some work we have lined up in the future. First, I'll start by talking about work we did in the past, which is using parents from Sweden, which is a high latitude population with low stamen loss, and Italy, a low latitude population with high stamen loss, so fewer stamens present, short stamens. And using those populations, there were 3 QTL identified for explaining this stamen number. So 1 QTL, so a potential locus at chromosome 1, a potential locus at chromosome 3, or loci, and 1 at chromosome 5. So these QTL were identified using QTL analyses, and from there we were able to make, other collaborators were able to make these near isogenic lines, these nils, with introgression at these stamen loss QTL. So we have plants from the Italian that are completely Italian background, or plants that are completely Swedish background, and then we have 3 single nils. So an Italian plant with introgression of just QTL1 for stamen loss using the Swedish genes, introgression at just QTL3, introgression at just QTL on chromosome 5, and then vice versa. So we have these nils where we have one background, but stamen loss genes from a different background. And what we're doing currently is making crosses to create double nils and eventually triple nils. So in the end, once all these crosses are done and we've looked at the genetic or the genotypes at these different loci, we'll have triple nils. So one plant that is entirely an Italian background, but there is Swedish genes at these 3 stamen loss QTL and vice versa. So these are going to be really cool and will help us find out more about the effects of the short stamen number on its own, independent of the rest of the genetic background. So we know already that these parent populations are locally adapted. So the Italian plants do better in Italy, the Swedish plants do better in Sweden. And our goal is to, once we have these double and triple nils all completely developed, is to do a reciprocal transplant. So we have the parent and nils present in field sites in each country. And then we'll look for fitness effects. And this will help us to isolate the effects of short stamen number alone, again, independent of the rest of the plant background, on fitness. So this is a really cool experiment we have in the pipeline that we're doing the crosses for currently, and we'll hopefully have triple nils pretty soon. Finally, we have a few more experiments going on in the lab that I'll mention quickly. One is fine mapping and candidate gene tests. So student Madison Plunkert, who she did a rotation in the Connor lab early on in her grad student career and is now still working with these QTLs. She's looking for genes underlying the major effect QTL on chromosome 5 using knockout lines, and then looking for shared SNP differences from the parents to identify the gene or genes at this QTL. And then finally, we have dry weight and elemental analysis planned. And this is to help us identify the resource cost of short stamens. So as I said, it's really hard to find evidence of costs in nature. So this has helped to pin down what the actual resource cost of producing short stamens could be. No work has been done on this yet, but something else in the pipeline. So with that, I want to acknowledge my whole lab, past and present, who did much of this work, collaborators, particularly Javier, who did so much to help us get this going, and currently, and then the NSF for funding, and thank you all for having me here. It's great to visit. Thank you.
Is this on? Sorry. Thank you so much, Jimmy. I have some questions, but I'll let other people if they have first.
I'm not a plant person, so my questions are like.
Yeah, go ahead.
Okay, I'll start with a really basic stuff. Everything I know, you just told me, right? But you mentioned, or I understood, that the long stamens and the short stamens had different amounts of pollen.
Yeah.
So do you have variation in the amount of pollen on the short stamens also, or only presence and absence, and they all have a high amount of pollen?
I don't think we have information on if there's variation in pollen number. I think it's just been found that on average, the short stamens produce more pollen than the long stamens. But it would be interesting to see if different populations, the short stamens produce more than others, and if that varied altitudinally. That's definitely, and latitudinally. So we don't have that information, but for sure it'd be interesting to.
So a few more super basic questions. So I study mammals that I know are really different from plants. But in the high elevation populations and species, the individual species are generally denser. Like there's more individuals of the same. Is it that way in yours? Are there?
I mean, they definitely grow smaller and more like tightly. The rosettes are smaller and more tightly clustered. I'm not sure about natural.
I mean, like individual plants.
Right. I don't actually know. I've never witnessed high altitude or high latitude plants in the wild, but they have more. Their rosettes are tight, more close to, their leaves are close together to help with dealing with frost and snow cover and stuff like that. So it would make sense if they were, but do you know?
Yeah, well, there's a huge diversity at, thank you. There's a huge diversity at high altitudes. It's not the same in northern Spain and southern Spain, right? So I recall huge plants up in the mountains as well.
Well, I mean, like the number of plants. Like, do you have like more plants next to each other, or is it more separate out?
You can, any combination you can think of, you'll find it out in the field, at high elevation and at low elevation.
It's
not
not associated.
No, not at all, no.
Okay.
These are pretty weedy plants. You kind of find them anywhere and everywhere.
So they're weedy everywhere.
No. Some of these populations are really wild populations, growing with a huge diversity of other plants.
Yeah.
Isolated next to each other, under a bush,
But that's not associated with elevation.
Oh no! It's the vegetation type and the environment, right? So here in the south, for example, when you are in the southern half of Iberia, you have to look under the shrubs and trees because the plant looks for cover, right? And for protection from insolation and heat. But if you move upwards, then you have to look the plant in the openings. Because when it's too shadow and too dark, so it depends on the place that you are. Everything changes, even the habitat of the plant. So that's no relationship there. I would say.
Okay, there was one more thing that you mentioned. You said that there was about 1%. Here on this slide, about 1% outcrossing rate, and I was wondering if there was a variance in that with habitat.
So it has been reported as we found different reports of outcrossing rate. It has varied in reports, but we don't actually know if it varies by altitude. We had a planned study to look for outcrossing rate, but logistically it's a lot to, since it's so low, it's a lot to put time into something we expect to be
answer
close
to
zero.
that. Actually, Hoffman's paper, it's a bit outdated already, because we know that this natural outcrossing rate is between 4-6%, right? And at high altitude, because you are losing genetic diversity, you are losing as well heterozygosity as well.
Yeah.
But there are populations in central Germany, for example, they found up to 14% of outcrossing rate. So depending on the year, in good years when you have carpets of plants, I've seen a bunch of pollinators visiting this plant, right? So for me it makes sense, to find on average between 4 and 6%. Actually, that paper comes from an experiment in a botanical garden, so it was quite unnatural.
Yeah.
About that. But in natural populations, between 4 and 6, even 8% of natural outcrossing, which is a lot.
Yeah.
For a sulfur, detecting this kind of outcrossing rates creates a huge amount of genetic variance and a huge amount of phenotypic variance as well. So, yeah.
So there's going to be selection in these populations with lower genetic diversity for more outcrossing?
Oh, absolutely. Actually, even with this sulfur, you detect the hybrid vigor.
Yeah.
So hybrids, perform better than the parental.
So the high elevation should be outcrossing more, and they should have those other stamens, right?
Well, at crossing rates, we haven't detected an altitudinal gradient in outcrossing, because again, it depends on the environment. And at high altitudes, the pollinator community is less diverse, less active. So, it's more difficult to detect this kind of rates up there.
Thank you. So I have a couple, I think we've got plenty of time still. I have a few questions. First one maybe is a bit of an annoying question, but how representative of fitness is seeds per fruit? So like, is it like, are there other ways that fitness could be changing across the life cycle, for example, like number of viable
Yes, so
seedlings or something?
it's not very representative. It's representative function, but not overall fitness. So the bigger difference in fitness we see is for total fruit number. There tends to be some variation of seed set per fruit, but the big thing is fruit number. So plants that are producing a ton of fruits versus plants that are producing, they make 10 or 11 flowers and they make 10 or 11 fruits. So that's where we see the bigger difference in fitness overall.
Okay, cool. But you don't necessarily, you haven't got that for the studies previously. You haven't got that like fitness measure relative to the short stamen.
Right.
number.
So that's what we're hoping to get this year.
Cool.
Yeah.
Awesome. Okay. Next question was, yes, in the original cline, in the original altitudinal cline that you saw with the nice coloured figure.
Oops, I went the wrong way.
I'm just curious about the shape of the relationship and what might be driving the kind of like, I don't know whether you would call it a downtick at the end, but like there is like, how you modeled it and like what kind of, what.
I think it's more representative of saturation. So once you get to close to 2 short statement numbers on average, we're not seeing much more of an incline past a certain elevation.
Cool. Okay.
So.
Yeah, so it's just kind of it's just like a general saturation.
Yeah, I couldn't really tell you what, but it's just a population that happens to be slightly lower there. But yeah, we tend to see it saturate after a point. But
Cool.
do you have anything to add to that?
Well, that's a very small population.
Yeah.
It's just a patch.
It's just a small patch.
In the middle of the middle, and I spent a lot of time looking for more patches, but the high altitude populations are quite tricky.
Yes.
They are super patchy.
I imagine. Yeah. Interesting. Oh, cool.
I should say, sorry, we do have fitness measurements from our chamber experiments, but we haven't, that hasn't come up in our, we haven't seen fitness differences yet, but we're hoping we'll see something else with our bigger set that's going on currently.
Awesome.
Yeah.
Awesome. Another slight question from the big line study that you showed, and kind of related to this as well, is it looks like there's a variance effect as well, like there's like a phenotypic variation effect as well. So like higher altitudes or higher latitudes, as well as having a more stamen loss. Is that right? Yeah. More stamen loss. You also have smaller
opposite.
variation in...
Opposite, yeah,
All the way around, sorry. More
stamen retention,
more
more
stamen
short
retention.
stamens. Yeah.
You also have smaller variation in stamen retention. I think it was particularly in the intensive set, it was particularly clear. Yes, this right side plot, there looks like there's quite a big variance effect. So like, yeah, on the right side, I don't know if you could, is that just highlighting the genetic diversity difference perhaps at different latitudes or altitudes, or is that, is there something else going on there? Like why is it, for example, that you still get a lot of short stamen numbers even at low latitudes, for example?
So one thing here, I've included both Spanish and Italian populations where we're seeing, these aren't just like strict altitudinal declines within a single mountain range or anything. So there's variation even in the lower latitude populations that have more loss on average. There still is a lot of variation between the populations in stamen number. But yeah, we do tend to see less variation in stamen number in the high latitude and altitude populations where there is just less stamen loss overall. But yeah, even in low latitude and altitude populations, we still find a good number of 2 short stamen flowers in general. So yeah, that could be due to less genetic variation at these higher populations, or it could be, again, some of the other mechanisms that are retaining.
Actually, I had a question about this slide and that panel in particular.
Yeah.
So perhaps you recall the discussions we had in the evolution paper in which, I was defending the idea that development can be key in this story. And I was really interested in seeing that flowering rate, right? Not flowering time, but flowering rate, so the speed at which the plant completes the cycle, which was the trait covering with the stamen loss. So I'm not surprised that only in Mediterranean populations you detect clear patterns of variation, just because the genetic diversity around here is higher, and even the demographic history is different, right, between the rest of Europe. But another, let's say, element supporting my hypothesis that development can be key here is that here in the Iberian Peninsula, all the populations above 800, 900 meters require vernalization to flower.
Yeah.
So if vernalization means that the plant, when it's in the vegetative stage, it needs to feel the cold of the winter. Okay. Right? So it's, this kind of cold hour counters that you need to accumulate cold and intensity of cold to then make the transition into reproductive. So when you are at high altitude, all the genotypes, if you grow them in the greenhouse, you have to move them back to the growth chamber and give them cold for 6-8 weeks. If not, they don't flower. So the whole thing changes when you are above this turning point. And again, the number of short stamens, there's a clear cut here as well. So I think that there's something related to the way the plant completes the cycle, and perhaps the genetic mechanisms that are required to complete the cycle. And if you don't have to vernalize, or just with a few days or few weeks of cold weather, like it's happening in southern Spain, it's enough to activate the whole mechanism using the same genes, by the way, right? But it doesn't happen at high altitude. There you need cold to complete the cycle. Yeah. So this pleiotropy thing, I think that, and flowering rate is something pointing to that direction.
Definitely.
There must be something related to all the genetic mechanisms underlying this vegetative to reproductive transition. Yeah. Right? So I enjoyed very much that flowering rate, not flowering time, is the one
Yeah, that's
here
what we found. It popped
telling
up.
something.
Yeah, we were surprised. We
Yeah.
didn't think about it before. We included flowering rate in our intensive set. We didn't have it in the broad set. We just looked
Absolutely.
at it and boom, it popped up.
Absolutely,
So, yeah.
absolutely. So, yeah. The other thing, if I may. Well, as you see, you have a small audience, but super interested in your stuff. So, just one curiosity. Are you going to do some sort of gene expression in your triple nils as well?
In our 3 nils,
Yes.
I'm not sure if that's planned currently.
Just to make sure, that's
oh, yeah.
eventually, the 3 QTLs, there's some sort of expression there
Yes,
going on.
we will be. Yeah, we'll be
yeah.
vetting our 3 QTLs and for our triple.
Well, that material is gold for this kind of thing.
Yeah.
So that's yeah. And in Arabidopsis, we share all materials,
Yeah.
which is something that that makes a difference.
Yeah.
With other research communities, we share everything.
Yeah.
After publication. So.
So we're excited to have those nils develop soon. We can.
That was going to be how I finished, was just to say like how refreshing it is to see such a large number of studies from the same species.
Yeah.
Like such a high number of application. It's really beautiful work. Yeah, and it's super, super cool. Like so many interesting things. So many interesting things to ask, but I think we're probably going to run out of time. So thank you very much. Thank you for coming, everyone who's
thank
come.
you all.
And we'll see you next week. Yeah. Thank you
Thank
very
you for
much.
having me.